everybody. Welcome to our mini lectures on artificial selections created by the one and only Group 5. So, why is artificial selection important or interesting to learn about in general? If you're going into a field that requires a knowledge in biology or, of course, here genetics, um, or even if you want to become a vet or learn more about the cultivation of plants and animals, it's important to have at least a little bit of knowledge, of course, about the artificial selections and its process. So today we're going to be talking a lot about artificial selections, but we're going to be also talking a little bit about natural selections and how it's different from artificial. Examples such as corn kernels, reptiles, the breeding of domestic canines, and watermelon seeds will be served as an example that will cover the molecular and the Mendelian genetics of artificial selections. So, what is the difference between natural selection and artificial selection? Of course, for artificial selections, the breeding is more controlled. Um, Breeders can choose a certain plant to mate with another plant or an animal to mate with another animal, and to breed a desirable trait and leaving out the ones that they don't want to be apparent in their offspring. Which is very different from natural selections because natural selections is more of free. It's based on the environmental conditions and the characteristics of these animals. They're based on the population's ability to adapt, survive, and reproduce, which of course is controlled by the environment and is very different from the artificial selection processes. And not to mention that natural selection is quite simple and straightforward because it's just nature. So, a little bit overview of the molecular genetics before we get into the Mendelian genetics and all the examples. Molecular genetics, there's a lot of things that you can learn from this aspect. Of course, with the technologies that we have now, a bunch of genomes and base pairs can be discovered when you artificial select these type of animals, and it's very beneficial when it comes to obtaining molecular footprints of ancestor and to learn a little bit more about their chromosomes and genome expressions. Another way it's the molecular genetics is very important in artificial selections is that it's able to discover the certain types of genotypes and phenotypes that is produced by artificial selection. Of course, these phenotypes are able to be found based on the marker-assisted selection, such as utilizing the genetic polymorphism, which is the marker loci, and it also improves the quantitative traits as well when it comes to studying these organisms.
About Mendelian genetics in regards to artificial selection, we need to remember that it's been going on since probably before Mendel did his experiments with the pea plants. And Mendel selectively bred pea plants in his experiment and focused on traits such as color of the petals, size of the plant, shape of the peas, and he also controlled the mating between female and male parts of the plants to reach the observations and conclusions that he obtained. Generally speaking, Mendelian genetics is the portion that most people are aware of and think about when genetics are talked about. And today we can really see Mendelian genetics being used in artificial selection in a variety of industries, such as agriculture and especially in pet breeding. Now we're going to talk about the human impact on artificial selection through various examples, starting with genetic manipulation in domesticating canines. To begin, I'm going to go over a Brief history. The origin of dogs starts with the domestication of wolves that occurred roughly 14,000 years ago. Hunters noticed that some wolves had lower temperament compared to their aggressive pack members and used it as an opportunity to interact with these wolves and build a relationship with them so they could aid hunters in hunting. And through this, they were also able to establish that man and canine companionship. Over time, wolves physically and genetically changed into the canines that we see today. And once genetics was researched more and popularized in science, a theory was created that stated the enhancement or change of one trait will have an effect on other traits as well. And with that in mind, Russian scientist Dmitry Belyaev and his colleagues decided to test that theory, and they experimented with foxes. Belyaev and his team focused on the fox's temperament by creating two separate breeding groups, where one breeding group was focused on enhancing aggression and the other was focused on lowering that temperament. Over time, they noticed physical and chemical differences in the offspring. The aggressive foxes had slightly darker coats, they were bigger in size, and their stress levels were higher due to their elevated alertness, whereas the low-tempered foxes had floppy ears, a drastic change in their coat, and reduced stress levels. So these observations really proved that the theory was correct. Over time, dogs have been bred for a variety of desired traits. The Quaker Houndhound was bred for hunting ducks. They can swim in water, and if trained effectively, they can use their tail to lure ducks to the hunters. Some dogs were bred specifically for size to make them small lapdog dogs, such as the Yorkshire Terrier, also known as the Yorkie. Another trait is mostly closely related to the aesthetics of the dog in the sense of the coat color, the coat patterns, size and shape, which is what we see nowadays in bully breeds. The rare colors and patterns make these dogs more valuable in the market. Additionally, dogs have been bred for their temperament, such as the Pekingese breed. They are known to have very low temperament. They're very relaxed and make great family dogs, which may be due to their part of their ancestry that leads back to when they were pets of Chinese emperors. And lastly, we have breeding for intelligence. So retrievers are a great example of this because they're known to be very intelligent and they're one of the easiest dogs to train, and they are commonly seen working as service dogs. On the other side of things, the molecular side, there's been information that's been discovered that relates to artificial selection, but there's also information outside of that, which is also important to artificial selection. It's been hypothesized that the wolves from 14,000 years ago had a very large capacity of diversity, which then resulted in the wide range of breeds that we have today, along with their wide range of traits. Additionally, certain molecular and genetic factors interacting in different positions can affect gene expression. For example, short interspersed nuclear elements, also known as SINEs, in certain positions of the DNA sequence can cause neurological effects like narcolepsy or phenotypic mutations like the Merle coat pattern as seen in the French bulldog in the previous slide. Another piece of information that's been discovered is that canine DNA is genetically closer to human DNA compared to that of mice. And scientists have said that this can really aid in medicinal research better than mice because of the fact that canine and human DNA are a lot more closely related. But the usage of canines in medicinal research has been refrained because of ethical issues and, of course, other reasons as well. In regards to breeding, molecular genetics of canines can help identify genes associated with non-Mendelian and polygenic traits. So this can help breeders better predict the quality of potential and future litters since they will have a better understanding of what is molecularly going on, such as when mutations occur.
One of our topics in the discussion of artificial selection will be the development of watermelon. The original ancestor of watermelons developed over 5,000 years ago in South Africa. These melons were very different than the ones today. They were a very bitter fruit with an extremely hard rind that had to be pried open with a pick and hammer. They were less than a fifth of the size of modern melons and were known for their ability to contain water. Much of what we know about them comes from Egyptian writings describing their cultivation and the remains of melons left in tombs of kings meant to feed them in the afterlife. Watermelon would soon become widespread throughout Africa in 2000 BC. The first recorded watermelon crop would be cultivated in Egypt. Not long after, it will begin to spread to other parts of the world through trade and eventually reaching America by the 16th century, making them a global crop. Ancient Egyptians originally cultivated watermelons for their unique ability to store water. They developed helped them thrive in their arid climate. They bred the watermelon to be capable of holding more water by taking seeds from the biggest melon from each crop and planting them in the next season. Europeans would soon obtain the fruit and begin crossbreeding the species with other melons using similar cultivation patterns, focusing on a sweeter and softer melon. Today, there exist over 1,000 varieties of watermelon due to crossbreeding and hybridization. Modern day watermelons are much sweeter and softer than their ancestor with a higher water content and can be over 5 times the size of the original watermelon. One of the most popular melons today is the seedless variety. Within the past 80 years, we have created hybrid watermelons that no longer contain seeds, which have become commercially valuable. The genetic makeup of modern day watermelons are very distinct from the original ancestor due to generations of selective breeding brought on by humans beginning as far back as ancient Egypt. Another topic in our discussion of artificial selection will be the development of corn or maize from a nearly unrecognizable plant into a yellow cob we know today. The original ancestor of corn existed nearly 10,000 years ago in the lowlands of Mexico. It was a wild grain known as teosinte. This grain was a dry, yellowish green with very few kernels in comparison to modern day sweet corn. The kernels of this plant were extremely hard and had to be cracked open like a nut or popped with heat. Many historians believe ancient Native Americans may have ate the kernels in a method similar to how we make popcorn today. The Native Americans were some of the first indigenous people to successfully cultivate the grain and introduce it into their society. The introduction of the grain to the first European settlers in the 16th century will lead to the grain spreading across the globe, which would eventually lead to many diverse strains of corn developing on different continents. Ancient civilizations would soon begin to practice artificial selection as they would realize not all plants share the same qualities and would purposely save the kernels from plants with desirable traits to be planted in the next season. Over generations, progressive changes begin to emerge in the crop, thus demonstrating an early understanding of the heritage of traits within plants. Corn, as a result, will progressively grow softer kernels in increasing numbers, allowing more corn to be produced per crop cycle. After generations of artificial selection and hybridization, we have went from a tiny stalk with few kernels to large sweet corn with hundreds of kernels we now in love today. Corn today have many desirable traits for consumption, repelling of insects, and drought resistance that wouldn't be possible without artificial selection. The genetic makeup of corn has changed drastically over a 10,000 year period due to human influence. Currently, there are 6 types of kernels worldwide, being flint, flour, dent, pop, sweet, and waxy corn, all thanks to artificial selection.
And I'm going to be talking about reptile pet trade morphs and colorations. Our first example here is the wild type versus the pied ball python. The piebald is a recessive trait that is responsible for partially unpigmented with variable color and pattern mutations. The piebald express a unique cloud-like pattern that is unseen in any other kind of base morph. Our first guy up top is the base morph. And he has just the yellows, the black color, and the tan color, whereas our guy on the bottom here, our lightning pied ball python, shows the cloud coloration with the pattern that is definitely spotted, and it looks like he's missing parts of his pattern when that's actually the piebald gene at play, but he is actually a lightning ball python, which exhibits the unique two codominant traits that are the piebald gene and the axanthism gene. And the axanthism gene is going to code for the lack of xanthophores, which are responsible for the yellow pigmentation in the top guy that this guy lacks. And that gives the lightning pied ball python more of a pastel color, which is really cool in the pet trade because it gives him a unique color. Something that's really important to note here about our morphs is that usually morphs would not stand a chance in the wild. These guys are only found in captivity, and the reason for that is that with these unique colors and obviously flashy colors, these guys would make great prey in the wild, and that's not something that you can survive long enough to reproduce with in the wild, but that does make them very cool pets and very desirable for pets. And our next example is green iguana morphs. Here we have our blue morph and our standard green common iguana. And it's important to note that there are over 10 different morphs of iguana, going anywhere from yellow to red morphs. Another cool thing about iguanas is that this guy here, Ivan, is not a red morph. He is actually just an aged iguana. He's about 16 years old, and these guys do change color as they age, but that's not necessarily because they are any kind of morph. That's just how they age. And our key takeaway here is that artificial selection impacts the world today, from the food that we eat to the pets we take care of, and artificial selection is different from natural selection, in that artificial selection is done deliberately, and organisms such as corn kernels, watermelon seeds, reptile morphs, and colorations, and domestic canines all are impacted by this, and there are Mendelian and molecular genetics in regards to artificial selections. The pros and the cons are that there are pros for farmers and production markets, and they enhance the quality of the product used in every day, no matter what the change. Cons is that there is an increase in diseases and the lifespan decreases, lessens the number of variations in the gene pool, which results in higher risk of mutation, which can also be considered animal cruelty. And there are some things to ask yourself. Is artificial selection worth it? And should we ever stop doing artificial selection? Is there anything that could stop us from doing that, or potential benefits to this? And with that, thank you so much for watching our presentation. And that, goodbye.